Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is related to the RNA processing. And RNA processing generally involves the first one is the capping, 5' capping, second one is the polyadenylation, and third one is the RNA editing. And splicing is another topic related with the RNA processing. So that will I will try to describe in another video. Here I will just try to explain about the polyadenylation. Okay, and if you are interested about the first part, that is the 5' capping, so you just check out the my channel and see it. Okay, and it is my request to the newcomers, who are just coming to see the videos here. You just share, subscribe my channel for the growth of this channel, and if this is helpful to you. Okay, so let's continue. That is the polyadenylation, and this is the polyadenylation is generally all eukaryotic mRNAs have a series of nucleotides are there, generally up to 250 adenosines present at their 3' ends. Okay, so what is saying that up to 250 adenosines present at the 3' ends, and this is called the poly-A tail. Okay, this is called the poly-A tail, and these are not the specified by DNA. Okay, these are not specified by DNA and are added to the transcript, that means mRNA, by the template independent RNA polymerase, and this is called the poly-A polymerase. So how this added with the transcript with the help of polymerase, and then it is known as the poly-A polymerase, but these are not identified by the DNA. Okay, or you can say the specified by the DNA. That a single processing complex undertakes both the cutting and the polyadenylation. Okay, here is a single processing complex, or you can say that one important complex which is responsible for the cutting and the polyadenylation of the transcript. Generally, in case of mammal, the mammal polyadenylations, that is one single sequence is 5' AAU AA3'. It is involved in the polyadenylation process, or you can say this is a single signal sequence which involves in the polyadenylation process in the mammals. And where it is located? It is located in 10 to 30 nucleotides upstream. Okay, 10 to 30 nucleotides upstream of polyadenylation site. There are two streams present in the transcript. One is the upstream and another is the downstream. So I think this is another topic, so here I not explain this about the upstream and the downstream, what is called the upstream and the downstream. You can see the transcription process in my previous video, or you just search out that the transcription process in the from my channel, so you can, I think you can get this the knowledge about the upstream and the downstream. Okay, so here, so what I am saying, that 10 to 30 nucleotides that present in the upstream of the polyadenylation site, and in the next one, that is the 10 to 20 nucleotides downstream of the polyadenylation site, is known as the GU reach region. So what is saying that in the 10 to 30 nucleotides upstream, here upstream of the polyadenylation site, there is present this 5' AAU AA3'. Okay, and this is signal, one kind of signal. And in the 10 to 20 nucleotides in the downstream, there the GU reach region. The two important thing is here must be involved in this polyadenylation process. This both poly-A signal and the GU reach are binding sites for multi-subunit protein complexes. Okay, multi-subunit protein complexes, and which are respectively the cleavage and polyadenylation specificity factor, this is called the CPSF, and the cleavage stimulation factor CSTF. So which subunits are important here? That means the protein subunit or protein complex important here. The first one is the CPSF and the second one is the CSTF. Okay, so and where they can bind at the poly A signal and obviously on the GU rich region. Okay, now the generation of the proper 3' terminal structure requires an endonuclease. Here one thing is very important, role played by the endonuclease. Okay, endonuclease, what is the role of the endonuclease? It helps to cleave the RNA. Okay, it helps to cleave the RNA, and within the endonuclease the components are very important, that is the cleavage factor 1, cleavage factor 2 and the GF2. Then cleavage is followed in the next step. The cleavage is followed by the polyadenylation. So after cleavage or cutting of the mRNA by the endonuclease, then the polyadenylation process becomes start. And this poly A polymerase catalyzes, that means this is the enzyme that poly A polymerase, this catalyzes the reaction. Which reaction? Polyadenylation reaction. And obviously here important role is played by the ATP, adenosine triphosphate. And in the first step, what happened in the polyadenylation process? A short oligo A sequence, that is 10 up to 10 residues that added to the 3' end. Okay, and then oligo A tail is extended, that becomes extended up to 200 residue length, and this is the polyadenylation process. Okay, first it is added and then it is extended. Okay, and this reaction requires another stimulatory factor that recognizes this oligo A tail. So where is important factor is necessary? This is the stimulatory factor which recognizes this oligo A tail, which must be added with the transcript at the 3' end. Clear? The polyadenylate binding protein, that is called the PABP in the short form, it helps the polymerase to add adenosines, influences the length of poly A tail, and that is synthesized and appears to play a role in maintenance of the poly A tail. So what is saying that the one kind of protein, this is called the polyadenylate binding protein, it helps the polymerase what? To add the adenosines, that means A, to add the adenosines. Okay, and also influences to increase the length of the poly A tail. Okay, and this, in that way the poly A tail is formed. So now what are the functions of the polyadenylation? First one, that mature mRNA is exported from the nucleus and affects the stability of mRNA. Obviously, it by the process of polyadenylation, the stability of the mRNA is maintained, and obviously serves as the recognition signal for binding of the translational factors during the initiation process, that is seen in the translation process. Obviously, translation process I have already described in my previous video, so you just check out what is the translation process, and obviously Orbel hypothesis and another related to the translation I have already explained in my channel, so you just check out. Okay, so here the polyadenylation process is described completely. I think you understood. Okay, and in the next video I will try to explain another topic, that is the splicing process, RNA splicing process. Okay, so you just continue with my channel, and by sharing and subscribing this channel, you just support me. Okay, and this is not only for the growth of this channel, that sharing and subscribing process, because I just think that if this channel is helpful, so another people also need this channel for getting the knowledge. So you just help that by sharing and subscribing to support me. Okay, thank you.